There are three components in a cell cycle: interphase, mitosis, and cytokinesis. Interphase has three parts: G1, S, and G2. There are checkpoints during G1, G2, and at the end of mitosis. There is no start or end to the cell cycle. Mitosis is the most important process in biology, as the cells will divide and grow, which maintain the growth and living of ant and plant. Phase, the duplication of centrosomes and chromosomes in the G2 and S phase. In prophase, the nuclei disappear, then duplicated chromosomes become two identical sister chromatids through cohesion and centromeres. Mitotic spindle fiber begin to form, and last centrosomes start to move apart. In the next stage, prometaphase, the nuclear envelope disappears and chromosomes become more condensed. Also, kinetochores start to form and attach to the microtubules. Here, in metaphase, the spindles form, the chromosome line up across the metaphase plate, an imaginary plane in the middle of the cell. Each chromosome is attached to a spindle fiber at its centromere. Anaphase, the shortest stage, starts when the centromeres of each chromosome separate. The microtubules pull the newly formed chromosomes to each side of the cell. The final stages of mitosis are telophase and cytokinesis. Telophase is when the nuclear envelope and nucleus start to form. This info will be identical to their mother cell. Cytokinesis is different for animals and plants. For animals, the cleavage furrow appears and pinches the cells in two. For plant cells, a cell plate forms and the cell divides it into two as well. Both cells will be identical to their mother cell. Meiosis is the division of sex cells. It plays such a key role in reproduction and evolution that it creates a diversity, but while still maintaining the consistency of a species. Meiosis is divided into meiosis I and meiosis II. Meiosis I is a process of separating homologous chromosomes. Meiosis II is a process of separating heterologous cells. Independent assortment is the crossing over that occurs in prophase. It exchanges genetic information and produces reincumbent chromosomes. Random fertilization is the diversified aligning of chromosomes in metaphase I. It increases the possibility of how many genetic different cells can be produced. In prophase I, chromosomes condense and loosely paired homologs connect to each other. This is called synapsis. Crossing over is also completed between non-sister chromatids. Chiasma are observed later in the stages. Metaphase I, pairs of homologs arrange at the metaphase plate, while kinetochore microtubules attach to the centrosomes of the chromatids. Anaphase I, cohesion breaks down and homologs move towards opposite poles, while sister chromatids remain attached. Telophase and cytokinesis I, two haploid daughter cells are formed. Each chromosome consists two sister chromatids. During prophase II, spindle apparatus forms while chromosomes move toward the metaphase plate to get lined up for metaphase II. Kinetochores also attach to microtubules from the poles, separating sister chromatids, pulling them to opposite poles as individual chromosomes. Telophase and cytokinesis II. The chromosomes decondense and nuclei appear. 4 haploid daughter cells with different genetic information form. Meiosis is finally complete. Compare and contrast. Mitosis replicates DNA once and divides once. Meiosis replicates once and divides twice. Mitosis is diploid and meiosis is haploid. Mitosis produces 2 daughter cells that are identical to the mother cell, while meiosis daughter cells are genetically different and the number of daughter cells each meiosis produces is 4.